Hi all, Dr. Clark here again, and today I'm going to demo out the cell cycle lab. So the cell cycle lab will be looking at mitosis. I'll be looking at mitosis in plant cells and animal cells. Okay, so I'll show you those. It's a lot of microscope work. It's fairly simple material, but we'll get it done. The other piece of the cell cycle lab is karyotyping a organism. And karyotyping organism means that you're going to bust open the cells and make the chromosomes spill out of the cell and then examine the chromosomes. And you're going to examine the chromosomes and kind of classify the chromosomes by where the centromere, so if you remember back from lecture, centromere is what holds the two chromosomes together, okay? And so the centromere can be in the center, it could be towards one end, etc. And so either the chromosome is going to be metrocentric, telecentric, acentric. So that just determines, or that just distinguishes where is the centromere in relationship to the arms or the P side, the petite end of the chromosome, and the Q side of the chromosome. Okay, so I'm going to demo real quick. I'm going to show you first how to do a chromosome splat, and then I'm going to show you the microscope work and give you some video of the cells and point out the different cells and the different stages of mitosis, and then we'll come back and I'll show you the chromosomes from the chromosome splat. The reason why I'm going to do the chromosome splat first is it takes a while to dry, so I'm just going to demo that out. So if you're going to do this in lab, you probably want to do the chromosome splat first, and then you can come back and look at the stuff after it's dried and the slide's ready to go. Okay, so as far as the chromosome splat goes, it's a pretty simple procedure. In this Eppendorf tube, I have some what are typically called HeLa cells, and HeLa cells are named that way from a lady named Henrietta Lack, who donated her cancer cells to science in the 50s. Okay? And because cancer is just uncontrolled growth, cell growth, well, companies have been growing her cancer cells since the 50s and selling them to labs. And so they grow the cells in a culture, and then they capture those cells, and then they make it so the cells stop growing by putting methanol on them, and it stops the growth of the cells. And then we can do things like splat the cells and look at the chromosomes, because as cells are growing, they're going to be in different stages of mitosis, and we're interested in cells that are in the metaphase stage of mitosis, because the chromosomes are lined up in the middle. You can splat them, they'll come out of the cell, and you can get a good idea at what size those chromosomes are, etc., and they're all condensed up. Okay, so that's what I'm going to do. So first thing I'm going to do is grab an eyedropper, and I'm going to suck up some of these cancer cells, and I'm going to drop it onto the slide. Now, in order to make sure the slide or the cells are going to splat, I'm going to put this slide and block, the block just holds it at an angle, and the paper towel on the ground, and then I'm going to stand on the table. Because I can't get this on video, because my video is not going to, you won't be able to see me, you won't be able to see the drop or anything like that. I'm going to do that and then bring it back and show you the dyeing method, and then how we prepare the slide after it's dyed and dried, and then we'll examine it for the chromosomes. Okay, so I'll be right back. Okay, so I'm back, and what you see here are two separate dyes. The slide that I dropped the cancer cells on is now dry, and it's a very simple procedure of just dipping it in to each solution for 1 second, bringing it back out, dip it back in, so you're going to do that twice. You don't need to wipe off the excess solution. You can just go straight from solution 1 or stain 1, which is orange, into stain 2, which is purple. And then once you've done that twice, now the material is ready to rinse off. Okay? Now, when you rinse off a slide, the key to rinsing off a slide, I'm going to bring you over to the sink, okay, and show you the key to rinsing off a slide is you do not want to spray where the material is that you dropped cancer cell on the slide, maybe in the middle. You don't want to hit that directly. So even though it's dried to the slide and everything like that, you do have a chance of washing or rinsing that material off. So when you spray a slide, you want to spray it at the top, and then just let the water run down and kind of rinse that extra dye off, okay, but not spraying the location where you dropped the cancer cells directly. Okay, once you've done that, you can just lay it on a paper towel and allow it to air dry, and then it'll be ready for microscope work. You'll want to put a cover slip on it before you use oil immersion so you don't get dye onto your oil immersion lens. But nonetheless, you'll be ready to go. Okay, so what I have here, okay, I have a onion root tip mitosis cell or slide up here, and that's got onion root tips that have been cross-sectioned and put on the slide, embedded onto the slide. Okay, so what I'm going to show you is the process by which you need to start finding cells in different stages of mitosis. Okay, so I'm going to show you on the computer screen while I do the microscope work. Okay, so hopefully you can see that well. This is again an onion root tip cell or slide or cross section, and you can see, I'll show you this in a second when I zoom in more, but what this is a cross section of the onion root tip, and up here the cells are elongating. Okay? So these cells are just growing and doing their normal function. But down here, and we'll go down even further, the cells are actually multiplying. The cells are going through mitosis. Remember, mitosis is just nuclear division for the purpose of cellular division. Okay, so I'm going to look down here in what we would determine as the growth region or the reproduction region. This up here is normal function. Okay? And I'll show you how elongated these cells are. So the plants in the region where they're multiplying are kind of short, and the nuclei are dividing, and the plants that are up in this region, the elongation region, or the normal function, which we would just call the G1 phase region, these cells up here, they're not going to reproduce, and they are just carrying out their normal function. All right, so let's go ahead and just move a little bit further down so you can see the cells that are going to be in that mitosis, or in the different mitosis phases. If I move all the way down, and I'll show you this here in a second too when I zoom in, you're going to see this region down here. This is called the root cap, and the root cap is going to have numerous dead cells, okay, cells that are not alive, and they just provide kind of a protective layer for the growing cells. Okay, and I'll show you that once we zoom in. So right now we're at 40 times your eyesight, looking at a cross section of a onion root tip. Okay, and I'm going to move to 100 times your eyesight, so you can see this. Well, first I'm going to look at the root tip so I can show you the dead cells in that region. Oh. You know what? I think I'm going to move to a different root tip just because there is some blurriness there. There's something on the slide or something. Okay, so if you look, you can see there are some cells in the root tip or the root cap that have nuclei, but there's a lot of cells that do not have nuclei, and that would mean that those cells are dead, and it's like, basically it's like scar tissue. It's just leftover cells that can harden and provide a protective layer, and that's important for a plant root because this plant root is going to be traveling through the ground, and this area is going to get damaged by rocks and things like that. So they have this protective cap that doesn't damage the growing cells or the reproducing cells. So that's where I want to look, is in this region here. This is where the cells are reproducing. Okay, and I have to zoom in to 400 times your eyesight. I'm going to turn the light up a little bit and find focus this region so you can get an idea of what we're looking at. So I'm going to move up just a little more, and there's quite a few cells in lots of different stages of mitosis. Okay? So remember, this is mitosis because these are tissue cells. These are somatic cells. They are not germline cells. We're not looking at pollen or ova. We're looking at root tips. So we're looking at normal skin cells or normal somatic cells. Okay? Now there's a couple things that I just want to make sure that you understand what you're looking at here. The nucleus is the dark spot, and then you can see that the cell walls go around that nuclei. So this is a single cell, and so each one of those dark spots is surrounded by a cell wall and a cell membrane, and that would be, the cell. So there's lots of cells. Remember when we were talking about plant cells are not, when they reproduce, they're connected to the original cell. So they don't break apart. They don't form a cleavage furrow. Instead, a new cell plate is formed. Okay? And I'll show you this in just a second. I'm probably going to move to oil immersion just because it's easier to demonstrate the differences in cells when you go to oil immersion. So I'm going to do that now so we can look at some of these cells and I can go through the different phases of mitosis. We might move back to 400 times your eyesight when we do some counts, because sometimes under oil immersion it's a little more difficult to do counts, but under oil immersion it's a lot easier to see individual cells and you can get an idea of what's going on. Okay, so oil immersion, 1,000 times your eyesight, looking at plant cells in an onion root tip. Okay, so we can let's look at this group of cells right here for now, okay? So what you can see is these cells that you can see the nucleus, but it looks kind of grayish and there's no black long strands in it like this one. Okay, this one has black long strands. Okay, these ones here are in interphase. Okay, now What stage of interphase? That's very hard to distinguish. Okay, so remember, interphase consists of 3 stages: the G1 phase, the S phase, and the G2 phase. So I don't know what they're in. My guess is they are, in different stages. This one looks like it just reproduced, so these ones are probably in G1. This one looks like it's starting to condense. This could be in G2 or the S phase. Okay, but nonetheless, these are in the S phase. One way that you can definitely tell that is you can't see individual chromosomes, so you know it's an interphase. The other reason you can tell is you can see a nucleolus, okay, the site where ribosomes are made. And if the cell is going through mitosis, then the nucleolus will be gone, and the nuclear envelope will start to break down. Okay? So if we look at this cell here, you can see condensed chromosomes. The nuclear envelope looks like it started to break down. There's no nucleolus. Okay? This phase right here is in prophase. Okay? Or this cell right here is in prophase. If I move it over just a little bit, you can see 2 more cells that are in prophase. You can see right there. And these 2 cells, the chromosomes are condensed, the nuclear envelope starting to dissolve. Those are prophase. Okay. After prophase, the cell is going to enter metaphase. Okay, so I'm going to show you a metaphase. Okay, if you look right here at this cell, okay, these chromosomes are lined up in the middle, okay, on the metaphase plate, or what sometimes is called the equatorial plane. Okay, and they're getting ready for those chromosomes to separate. Okay? And remember that when they separate in mitosis, just one copy of the chromosome is going to one pole, and the other copy of the chromosome is going to the other pole. Okay? Now, if we're from metaphase, we're interested in the next phase, which would be anaphase. This cell here is in the early stages of anaphase. You can start to see that the migration is starting to happen to the poles. There's starting to be a little bit of white space here. So this is starting early anaphase. And that's one of the key components that I want you guys to understand, is that phases of mitosis are not cut and dry. So it's not just in metaphase, or it's not just in anaphase or telophase. It can be in the late stages of anaphase, early stages of metaphase, etc. So it's a continuous phase. It's not like cut and dry. And so you're going to see a lot of cells that are like halfway in metaphase and partway in telophase, or partway in anaphase. All right, so let me see if I can find a anaphase that is a little more noticeable. Before we do that, just because it's here, here's a good example of telophase. So if you notice there, you can see that the chromosomes are still condensed, and you can see the meiotic spindles here. You can see the start of a cell plate that's going to divide these cells in half. The chromosomes are still condensed, so you know that this is not cytokinesis. So in cytokinesis, you would have a nuclear envelope that wraps around these, and the chromosomes would uncondense. Okay, because cytokinesis is its own phase, and cytokinesis is technically the division of cytoplasm, or the final division of the cell. Mitosis is nuclear division. Okay, so you can have mitosis without cytokinesis. So you could have your nucleus divide in a cell without the cell dividing. Okay, it would result in 2 nuclei. This has happened and it does happen. Okay, but you can see here, meiotic spindles here in the middle. Okay, they're starting to dissolve, or they will start to dissolve. The nucleus will form on each end of the pole, and then a new cell plate will divide those 2 cells. Okay, so a good example of telophase. Here's another example of telophase. I'm trying to find a good example of anaphase. That one's early migration. Another good example of telophase here. Okay, so here's one right here. So there you can see these chromosomes used to be lined up on the middle, on the metaphase plate, equatorial plane, and now they're starting to migrate to the poles. And you can see that they're being dragged in between here. You can see the meiotic spindles, okay? And so those chromosomes are being separated, or the copies of the chromosomes are being separated. So remember that mitosis is the doubling of chromosomes in the synthesis phase. So when a cell is going through the cell cycle, it'll go from the G1 phase, where it's growing, to the S phase, where it copies all of its DNA. So in our case, we'd go from 46 chromosomes to 92. Okay? Then it goes to the G2 phase, where it's going to grow more, it's going to copy its mitochondria and other things that are needed for the cell to survive. All right, well, I'm back, and for this portion of the lab, if you're doing it virtually, you're going to examine the material that I'm going to provide you right now by looking at the different cells and determining what stage they are in. Now remember, we're only interested in the stages of mitosis, so we're only interested in prophase, metaphase, anaphase, and telophase. And you're only going to go until you reach 50 cells when those 4 stages are added together, that you reach 50, okay? So basically what you're trying to do is calculate, well, how often out of 50, or what's the frequency that cells are in prophase, what's the frequency that cells are in anaphase, metaphase, telophase. Okay, so I'm going to just move the slide, and this is going to pick up on the video, so you can start counting the cells in the different stages. Do not count interphase, because the majority of cells are in interphase. Okay? So, and interphase is not part of mitosis. Okay? All right, so I'm going to go ahead and start that. So I'm just going to migrate this slide. Okay, so at this point you should have easily got 50 cells in the different, or collectively in the 4 different stages. And we'll move on to examine animal mitosis. Alright, I'm back, and now we're examining whitefish, which is a type of fish, mitosis. So we're examining skin cells. These are actually cells, early cells within a whitefish's development. So basically after sperm and egg have come together, and this is called a blastula cell. So it's like the early tissue types, and we can start to examine these cells. So they're going to be rapidly reproducing, and then we can dye those and capture and see what phases of mitosis the cells are in. So that's why they occur in kind of these clumps. And remember that normally animal cells will divide through cytokinesis and they'll be separate cells. And you're going to see that still because these clumps are actually not connected to each other like a plant. So the plant is connected by a cell wall. These guys are actually free of each other. They just happen to occur in a cluster. Okay? Well, you're not going to see anything until we zoom in. So this is 40 times your eyesight. That is 100 times your eyesight. $400 times your eyesight. All right, so I'm gonna scan around until I find some cells that are easy for you to distinguish. There's going to be some phases that you already know and that you're fairly well rehearsed with. For example, over here, this is Anaphase. So what we're looking at, so you can see here a lot of these cells. This is a cell membrane. It's not a rigid cell wall, so this is a cell membrane. This is a nuclear envelope, and this cell would be in prophase. Because, well, interphase / prophase. You can start to see the chromosomes condense. You can see these dark spots condensing, so that would show you that it's starting to migrate into prophase. Same thing here, here, here. Okay, so there's quite a few. Now, if you look here, though, you don't see a nuclear envelope anymore, and you see meiotic spindles. Okay? And I'll go to oil immersion, so this is a little easier to see. But you see these meiotic spindles, and you can see these dark, kind of long projections. Those are the chromosomes. So there's chromosomes over here. There's chromosomes over here. They've been separated. This is anaphase moving into telophase. Now, telophase in animal cells is distinguished by the formation of a cleavage furrow. And that's where the cells are starting to pinch off. Here's a great example of that. So you can see these chromosomes here and chromosomes here, and then you can start to see the cell is pinching off there and the cell is pinching off there. So you can see this line starting to form between these two cells. This isn't completely pinched off and separated, because you can also see the meiotic spindles still forming between those two cells. Okay? So I'm going to go to oil immersion. I'll bring you back and I'll try to show you the 4 stages plus interphase and cytokinesis. Okay, we're back, and now we're under oil immersion, 1,000 times your eyesight, and again we're looking at blastula cells from whitefish. So these are animal cells that are going through mitosis. So here is that where we left off. This is that cell that's in telophase and is starting to separate. So it's pretty late in telophase because the cleavage furrow is almost complete, and these cells would be pinched off. You can still see that meiotic spindle connecting the two cells. So we're going to move over just a little bit. And here you can see a cell that's in the late stages of anaphase. You can see the chromosomes here and here. These are the poles of the cell. This is where the centromere is. Remember, in animal cells, centromeres is where the meiotic spindle comes out of, or the microtubules are formed from. So you can see these spindles. That's going to be elongating the cell, and eventually it'll move into telophase, would have, okay, moved into telophase and pinch off. All right, let's see if we can find a cell in metaphase. A lot of cells in prophase. You can see there. Oh, there's a metaphase. Okay, so here's metaphase next to prophase. So you can see here chromosomes lined up on the metaphase plate, equatorial plane. There's a centromere. There's a centromere. These are microtubules that are connecting to the centromere. So the microtubules are tipped with kinetochores. So the end of the microtubule has what we call a kinetochore. That attaches to the centromere of the chromosome and then starts to pull them apart. We use the term pull, but in actuality the microtubule is dissolved. So the chromosome walks itself kind of back to the pole. But at any rate, you can see here, this is metaphase. These cells are in prophase because you can start to see those chromosomes that you would see here condense. The nuclear envelope is starting to dissolve a little bit in both those, and that kind of gives you an idea of what's going on. If we just migrate back over, interphase is pretty easy to see. Well, I shouldn't say it's easy to see, but cells like this cell here, this is an interphase. You cannot see any, this is the nucleus, you cannot see any chromosomes. So there's no chromosomes condensed, etc. So that's the interphase. It's either in the G1S or G2 phase. Okay, so hopefully that's enough of the cells that you can get an idea of how to tell the difference between cells in interphase, prophase, metaphase, anaphase, and telophase, no matter whether we're dealing with plant cells or animal cells. When I come back next, we're going to examine the karyotype, or the chromosome splat that we made earlier. Okay, so I'm back, and now we're going to examine that karyotype slide, or the chromosome splat slide that we made earlier. Now the key when you're making a slide like this, remember you're looking for chromosomes that have bursted out of a cell, and as you could see from examining mitosis in plants and mitosis in animals, chromosomes are really small, and you're not going to be able to see them until you zoom in quite a bit. So my suggestion is, when you're trying to do this, is find something that has died on your slide, okay? Something that dyed purple in this case, because that's the dye that we're interested in. This is, the screen is not doing it really credit, but these little dots that you see there, these are all purple, okay? And so what I'm going to do is, now that I have some dots and I know that these are cells, and I actually think over here there might be a patch of chromosomes that is split open, I'm going to focus in on a single cell, and then I have my focus frame, I have it once I get to oil immersion. If I'm oil immersion onto that cell, then I know that I'm on the right depth of field to examine this slide. Okay, and that's kind of key, is once you're on the right depth of field, then you can go across the entire slide looking for areas where you might see cells that had burst, and you can count the chromosomes or examine the chromosomes. Okay, so I'm going to go ahead and move through fairly quickly. I'm going to move through and focus in on one of these cells and see what I can find. Okay. Okay, so now I'm moving to 400 times eyesight and increase the light a little bit. So there you can see that I have a cell right there at 400 times your eyesight, and I'm going to go to oil immersion so I can see if there's chromosomes nearby. So remember, when you're trying to focus a microscope and you're trying to find objects, you need to start at the lowest magnification and work your way up. That's the best method, and the quickest method is to start at 40 times your eyesight or scanning objective lens, and then start moving up until you get to the point where either you're in oil immersion, because that's what you need to be at in order to see the cells, okay, or whatever magnification you need to see that. So what I thought was just a single cell is actually two cells stuck together, but nonetheless we're in the right depth of field, the right reading frame for the slide, and now I can start examining nearby features to see if I can see chromosomes. And not too far away is a little patch of chromosomes that I just lost, but I will find them again. Okay, so here are some chromosomes. The cell has been busted open, and there you can start to see chromosomes. You can see this is the P arm of this chromosome and the Q arm. The P arm, known as the petite arm, sorry about that, is smaller than the Q arm. Okay, and you can see the centromere is right there, or where the material is pinched off. So, from this chromosome, or from the diagram that I give you on page 89, you might state that this is acrocentric or at least sub-metacentric. So you notice that this is not in the center. Okay, you can see that it's pinched right there. Here, same kind of thing. This is the P arm. These are the Q arms. Okay, again, sub-metacentric, metacentric, sub-metacentric, okay, sub-metacentric. This one looks to be metacentric. Looks like the P and the Q are about the exact same. Same over here. Okay, P arm, Q arm, about the same length. Okay, so that's metacentric. Okay. Either acrocentric, I think that's what that is. This one's a little harder to tell. Submetrocentric, submetrocentric. Okay, so I'm going to go through and find some more clumps of chromosomes and try to focus them just a little better. It's hard to see. So these are human chromosomes, and our chromosomes are pretty small, so it's kind of difficult to see them. But nonetheless, you'll see once you've looked at a lot of chromosomes, it's fairly easy to classify them by where the placement of their centromere. Okay, so I'm just going to go through, see if I can find some more chromosomes, and then I'll stop at those chromosomes, and you can get you can classify them once I stop there. Okay, another small patch of chromosomes, 3 chromosomes there. I'll let you classify them. Okay, it looks like there's 3 here. It's kind of difficult. These 2 are kind of merged together. They're overlapping with each other. Try to find another patch that's a little better. It's kind of difficult sometimes. You can see there's lots of cells in here that did not rupture, so even dropping the drops of solution from, I dropped them from about 8 feet up, it still wasn't enough to rupture some of these cells, so they're still whole. And some of the cells are not, they weren't in a phase in which the chromosomes are condensed anyways, so it doesn't really matter if they ruptured or not, because you wouldn't have got any data from them anyways. Here's a group of chromosomes there, so you can classify those. Trying to get this to focus nicely. It looks like there's 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 or so chromosomes there. Now some of them you won't be able to, like I said before, you won't be able to classify them because it looks like they got 6 or 7 arms to them, and that's because you have chromosomes stacked on top of chromosomes. Alright, so I'm going to find a couple more patches, and then the karyotyping will be over. And this is the method that I'm showing you here, demoed here. Here's another patch. It's the same method that we used to use in the 60s, 70s, 80s, even 50s, I think, maybe even before that, to determine how many chromosomes an organism had. So you'd splat them, put them on a microscope, take a picture, blow that picture up, cut the chromosomes out, match the chromosomes, and then count basically how many chromosomes or how many pairs they have, and whether or not the organism was diploid, so meaning they have 2 copies of every chromosome, triploid, 3 copies of every chromosome, tetraploid, etc. So this is a way we really found out that there's a lot of diversity throughout the animal kingdom, plant kingdom, all of them, with the number of chromosomes and how many copies of chromosomes, the sizes of chromosomes, etc. Today, it's much easier. It's much more digitized. Okay, a drop is still used, but it's slammed between 2 slides, splats them. The computer looks for things that are the exact same size and shape, matches them up, and then basically tells you what the number of chromosomes and how many pairs there are, etc. Now, cancer is not one that you really want to do this with if you were trying to karyotype an organism to find out how many chromosomes that organism has, cancer is not a good, cell type or cancerous cells are not good cell types to do that with, because cancerous cells are often they're mutated, and so you can have varying degrees of differences in those cells. So here's a bunch of chromosomes, and so we'll probably stop with that patch. So I'll allow you to go through. By now you've easily analyzed 10 chromosomes, and you have the ability to decide whether it's metrocentric, whether it's submetrocentric, whether it's acrocentric, or whether it's telocentric. Okay, and so that's generally how a karyotype is done, or used to be done, and that ends the cell cycle lab. Till next time.